All right. Well, thank you so much. Oh, excellent. Okay. Well, thank you so much, Benjamin. I've had a bunch of different different adventures in applied math and computational biology, and because the title of this seminar series here is the Biodynamics Days, I thought it would be prudent to talk a little bit about something dynamical. Now, this was work that I did, a very short project, honestly, that I scrambled together a few years ago about dolphin time series analysis. So let me try and explain what the main goal was. So the idea, the big pitch, that kind of goes beyond dolphins here, is the kind of larger question of, is it possible to somehow deduce or infer causal interactions? Hopefully, people can see the screen here. Some kind of information about what what biomarkers that you could read from a human have causal effects on other biomarkers. So, what is a biomarker? An example of a biomarker, a clinically accessible biomarker, would be something like body weight or the level of cholesterol in your blood or your red blood cell count. These are all biomarkers, something that you can measure from somebody's body that are easily accessible at the level of a clinic and that have some indication about your overall health or response to certain kinds of drugs or risk factors when it comes to certain diseases, etc. Right? So one of the things that clinicians have wanted for a long time is a way to kind of get some handle on, some predictive model for whether certain biomarkers or fluctuations in those biomarkers affect other biomarkers. And one of the big applications, one of the big I don't know, holy grails of medicine is to not only figure out which biomarkers affect which other biomarkers, but also how those effects change as people age. So questions regarding healthy aging, longevity, protection against age-related diseases, all of those things are of some interest to the National Institute of Health. And and so the question of how do biomarkers affect one another is ultimately supposed to help understand how people age. Okay? The trouble is, setting up an aging study in actual humans is really hard because there's so many different environmental effects that influence how people age and how their biomarkers manifest. So, for instance, a study in humans is going to involve lots of different humans with different diets, different socioeconomic backgrounds, different incomes, different medication histories, and so forth. And it's really hard to control for all those things. So, the hope was maybe we could learn something about the causal relationship between biomarkers and also hopefully learn something about aging by looking not at humans, but at some other long-lived mammal. Now, what other long-lived mammals are there, right? There's elephants, there's chimpanzees, there's some monkeys, perhaps. There are dolphins, but there aren't too many other long-lived mammals that have been the subject of very long-term, kind of longitudinal studies that could in any way assess something like aging or causal relationships between biomarkers. And we were fortunate enough to have access to this kind of one of a kind dolphin study. I'm going to go out on a limb. I don't know for sure, but I'm pretty sure this is this is one of very few. Like there may be one or two studies like this in the whole world, which is a study of a long-lived mammal, in this case a dolphin, which is a longitudinal study involving many, many years of collecting biomarkers across many, many animals. And this particular study was actually conducted by the US Navy over the last, 2 or 3 decades. And the US Navy has a dolphin research branch, which is kind of fascinating, right? And they train these dolphins and they work with them and they have the dolphins kind of play in like a sequestered area of the water. Like it's like a not a farm exactly, but like I don't know, a cordoned off area. And so they're able to control their diet and control their health. And so just as an example, you can of an example of the differences between these US Navy dolphins and what you might find in the wild is that a wild dolphin, a wild bottlenose dolphin, has an average lifespan of about 20 years. But the Navy dolphins, how many were there? There were actually 144 total, but this study only included 136. Um, Out of the Navy dolphins, their lifespan is significantly longer, like, more than 50% longer than the wild type dolphin lifespan on average. And some of these dolphins live to be, 45, 50 years old. And so what the Navy did is they would record these dolphins regularly over the course of 25 years. And one interesting kind of aspect to this whole study, which I'll bring up later, is that the Navy, their paramount concern was maintaining a good relationship with these dolphins. So when they would measure these dolphins, they didn't forcibly measure the dolphins regularly. They didn't measure every dolphin twice a week or anything like that. They would only measure the dolphins when the dolphins wanted to be measured. And of course, they had a variety of different measurements, body weight, et cetera, et cetera. And so they would only draw the dolphin's blood and measure things like red blood cell count and cholesterol levels when the dolphins were kind of happy to be measured. And so as a result, one of the idiosyncrasies of this data set is that the time points that the dolphins are measured over are not at all regular. Some dolphins, were, happy to be measured multiple times a day. Other dolphins were a little bit more grumpy and only wanted to be measured once every couple of months, etc. So anyway, well, we can get into that later. Now, what was our goal? Our goal was to take these measurements and try and figure out, is there any indication of any causal relationship between the biomarkers involved? But there's obviously a big elephant in the room. What's the elephant in the room? Which is that the body is super complicated. And what is a biomarker? A biomarker, something like red blood cell count, is like a very, very low dimensional projection of an otherwise very complicated high dimensional dynamical system, which is the body's regulatory system. So any particular biomarker can only ever give you a glimpse into what's going on and is going to be influenced by a whole bunch of unmeasured factors that you don't see. So what do we expect? What's the best thing we can possibly expect when looking at this kind of data is that, okay, if we look at some biomarkers in the body, what we might hope for in the best case scenario is there might be some kind of deterministic interaction between those variables, but there should also be lots and lots of something that looks like noise, some kind of shared variation or extra variation coming from all the unobserved things that we didn't measure. So just to summarize, what we expect is that we expect our time series analysis to involve, at best, this would be like the best case scenario, some kind of deterministic interactions that we can infer. That would be the causality that we're trying to find. But we should also expect to have to work around, or maybe also infer, some sort of stochastic drive, some kind of something that looks like noise coming from all the unobserved variables that we're not measuring. And there's an extra source of variation, which is measurement noise or observation noise, which I'll talk about in a second. And what's the punchline? The summary, just to jump to the conclusion, the summary here is that in this data set, even though it's a very, very well curated data set, probably, the best longitudinal study of biomarkers available for long-lived mammals that we have access to. Even though it's a really good data set, we still find for this data set that the kind of what looks like noise, the accumulated influence of all these unobserved biomarkers that we haven't seen, this noise is kind of the largest source of variation in the data set. And observation errors are also large. And while we can infer certain deterministic interactions, those are not the major role player in what's going on. These are more subtle. Okay, so that's the punchline. Okay, but before we get any further, let me give you a glimpse into the data set. So here's a figure illustrating what I think is a key aspect, an important feature of the data set. So what have I done here? I've taken one of the biomarkers. What is this biomarker? Alkaline phosphatase. This is a measure of a particular chemical in the blood. And if you go to the doctor and get your blood drawn, for a physical exam, um, that physical exam will measure a whole bunch of biomarkers from you. And it's that kind of physical exam that the U.S. Navy did on these dolphins, right? So when you go to the doctor and you get your blood drawn, the doctor measures your red blood cell count, your white blood cell count, your cholesterol levels, your iron levels, how much hemoglobin might be in your red blood cells, your alkaline phosphatase levels, and so forth. So that typical blood-based biomarker assay is what was done on these dolphins, and alkaline phosphatase was one of these biomarkers. And what I've done here is I've plotted the data collected across all 136 dolphins. And every point here represents what? Represents an increment between two measurements of a particular dolphin. So the horizontal axis is the square root of the time step between the previous measurement and the subsequent measurement. And the vertical value is the increment in the alkaline phosphatase level. So I'm plotting here the changes in alkaline phosphatase, vertical, versus the changes in time, horizontal. And the scatter plot here is the data collected across all the dolphins. And this scatter plot in the back, these black dots, is kind of a mess. There's a lot of overlapping signal. It's hard to see what's going on. So what I've done is I've binned this in time. So I've divided this into kind of vertical strips for various time bins, as you can see over here. And I've turned this scatter plot into a heat map, into a histogram. So every vertical slice is a distribution of variable increments for a particular time increment. And the color is representing the distribution here is pink for high and blue for low. And you can kind of see that these are unimodal, somewhat Gaussian-like distributions where the variance in the distribution increases. And here, what I've plotted, what are these white dots? This is the mean of the distribution at every time increment. And the yellow is the standard deviation of the time of the distribution for each time increment. And the key that I'm sure you've already recognized, but the key here is that the standard deviation seems to increase not exactly linearly, but roughly linearly up until the end where there's not many points, right? So this is kind of noisy at the end. But when you have a lot of data, you see that the standard deviation increases linearly with square root of the time increment. Put another way, the variance in the variable increments that you see increases linearly with the time increment dt. And that is pretty stark, right? It's pretty what do you call it? Clear that there is that trend in this data. And that's the kind of trend that you would expect if the increments were driven by Brownian motion, right? And we can look at another variable here. This is cholesterol. We can kind of see the same trend. Remember, the signal to noise ratio is kind of not so great towards the end here. There aren't that many points, so maybe you don't trust this stuff as much as you trust the beginning. And what you can see, again, same trend, that the variance scales linearly with the time increment, except it's not, how do you say? It's not linear through the origin. It's an affine linear function, right? Because this is the second important thing that I wanted to mention. If you look at time increment 0 here, the variance is still non-zero. Right? Let's go back here. See, time increment 0, the variance is non-zero. So this is telling you something else about the data. What's it telling you? It's telling you that there are a whole bunch of measurements here, right? All these dots here at 0. What they correspond to is the same dolphin being measured twice in the same day. And if the same dolphin is measured twice in the same day, you may not get the same result, right? And there are many reasons for that. It's possible that there's just measurement error. That, you measure the dolphin's temperature and there's a bit of either human error or there's error in the thermometer or there's some kind of error in the reading. And so there's some measurement error. So every time you measure the same dolphin, you get a different result, even if the dolphin's temperature hasn't changed. That's one possible source of this 0 time increment variation. There's another possible source too, which is that here, zero time increment really means you measure the same dolphin twice during the same day. So maybe you measure the dolphin both before and after lunch, or you measure the dolphin once in the morning and once at night. And the overall time scale of these measurements, kind of the canonical time scale, is maybe like weeks or months, right? Because the whole study spans years. The typical time scale between measurements might be several days. So when we say time step zero, what we really mean is a time step small enough that it's like much smaller than a week. And any fluctuation in the biomarkers that is very rapid with respect to a day or a week is something that we wouldn't expect to capture, right? So these time step zero variations might actually be due to just rapid fluctuations in the biomarker of a dolphin, right? Body temperature, blood sugar levels, these kinds of things. Okay, so that's the main observation that I wanted to get across. And then the last thing that I wanted to show is that there are certain biomarkers here. This is the number of segmented cells of a particular type in the blood that are, yeah, there's again some kind of plausibly linear relationship between time step and variation, but that Brownian component is much, much smaller than the static kind of noise in the observation that you would see at time step zero. So the baseline variation can be very large and and be even larger than the Brownian component. Or you can have a scenario like this where maybe the baseline variation is a little bit smaller than the Brownian component, or something like this. I guess this is also similar, right? Maybe the baseline variation and the Brownian component are of comparable strength. Okay. So um, that's the observation that motivates the following model, right? Because now when we look at this data, we can say, okay, there's all sorts of ways we can try and model this time series data. Why don't we try and model the time series data so that we have the opportunity to capture certain kinds of interactions? And let's just model them as linear interactions. I'll show you the equation in a second. But we should also, as a key part of the model, we should also build into our modeling of this time series data that there should be some opportunity for something like a Brownian process driving the variables involved. And there should be some kind of measurement noise that we should allow there to be variation even when the time step is zero. So what kind of a model could we have? Here's kind of the simplest model you could possibly write down that has those features. So just to summarize so far, taking the data, looking at how the increments of the data are distributed, that very, very, straightforward sanity check implies that any model for how this data works dynamically must include something like an interaction, because that's what we want to find, something like a Brownian process, because that's this, whoops, where did this go? Nope, didn't want to hit that, because that's this linear component here, and something like baseline variation, because that's the offset that we see here at time zero. And so a very simple model that does that is something like this stochastic differential equation. This is like, what do you guys call this thing? It's like a Ornstein-Uhlenbeck, I think. Maybe it's like a Langevin process with the quadratic potential, right? It's something like this, right? So let's imagine that we have some subset, let's say some subset D of these biomarkers. Let's represent that as a vector, capital X. And we imagine that those biomarkers are changing over time. And we expect to model those biomarkers as a very simple linear SDE. And that linear SDE is going to have some drift that we don't really care about. It's going to have some interaction component. This capital A is a matrix indicating the deterministic interactions between one biomarker and another. And that it's deterministic because this is the DT component, right? This is the change that is directly related to the previous measurement of the variables. And there's also some kind of a stochastic component here, right? There's going to be some kind of variation due to all the unobserved stuff that looks like noise. And what is capital B here? Capital B allows for the stochastic drive to be correlated between variables. And there's one part that I haven't mentioned so far, right? The part that I haven't mentioned so far is the baseline variation. What's the baseline variation? How is that captured here? And the baseline variation is captured in this model because we assume that while there is some underlying true biomarker, X, maybe we don't observe X. Maybe we observe some kind of time-smeared, regularized, noisy, who knows, version of X. Maybe we observe Y. And Y is X plus some observation noise. And as we said before, this observation noise, here epsilon is drawn from a Gaussian and C is the inverse covariance matrix of the observation noise. We assume that, again, this observation noise may be due to measurement error, human error, rapid fluctuations that are much faster than the time scale over which we are building our model, that kind of a thing. And so that's the simple model that we're going to fit to this data. And now for just some quick technical details here. Uh, should I mention these technical details? No, maybe I won't mention the technical details. I'll show you the results for now. Okay, so let's just jump to what this might look like. So here I've taken two biomarkers, iron, this is iron level in the blood, and MCH. What is MCH? It's mean corpuscular hemoglobin. So what does that mean? That means it's the average level of hemoglobin in your red blood cells. Some of your red blood cells have hemoglobin, some of your red blood cells have less hemoglobin or no hemoglobin. And hemoglobin, as you probably know, it's a molecule that you need to carry oxygen, and it's a molecule that itself uses iron. So if you're iron deficient, you have a harder time producing hemoglobin. Okay. And so what we're showing here over on this side are trajectories after Kalman filtering of the iron and hemoglobin measurements in various dolphins. So this is dolphin number 4, this is dolphin number 12, this is dolphin number 26. The color scale here represents the age of the dolphin from 0 to 55 years. So dolphin number 4 was initially measured when they were like, I don't know, 30 years old and measured towards the end of their life between ages 30 and 45. And you can kind of see this meandering, noisy spiral, I guess you could say, or noisy ellipse, noisy elliptical trajectory between iron and hemoglobin for dolphin 4. Dolphin 12 is a little bit noisier. Dolphin 26, you can see dolphins 12, 26, and 36 were measured when they were a little bit younger from, their when they were calves, 1 or 2 years old, up to about teenagers, 15, 20, etc. But again, you can kind of see the same kind of noisy elliptical trajectory. And this is only for the dolphins. If you take all 136 dolphins, and look at all of their trajectories, you again see this noisy elliptical thing more often than not. So what you can do is you can pool all the dolphins and fit this model to the dolphins, right? Allowing for some interaction, deterministic interaction, some noise, and some, where did it go? Where's C? And some observation noise, some Brownian stochastic drive and some observation noise. And if you do that, you get this kind of Ornstein-Uhlenbeck or stochastic differential equation shown over here. So here in the background, I'm showing the deterministic component. This is the capital A, x dt, giving rise to this slow this slow elliptical trajectory where iron and hemoglobin are influence one another, right? When the iron goes up, then the hemoglobin goes up, but as hemoglobin production goes up, the iron goes back down, and when the iron goes down, the hemoglobin production goes down, and so forth. So that's the inferred causal interaction between these two biomarkers that we can deduce from the data. And what's shown here, this squiggly thing, is just one kind of sample OU trajectory that accounts also for the Brownian Brownian drive that we can deduce for this example as well. What is the Brownian drive? That's actually not shown here, right? What I showed in the background here is just the capital A. It's the deterministic component. So let's take one step forward. And what I'm showing here, I don't know if you can see this. Maybe I can I, does this work? Maybe I can just zoom in here. That didn't work. Okay, never mind. I won't zoom in. Okay, so what am I showing here? What I'm showing here is for the same data across all the dolphins, I'm showing a scatter plot of the increments when of the variable increments in iron horizontal and mean corpuscular hemoglobin vertical when the time step is really small. So when the time step is really small, like basically zero, you see this distribution. This looks kind of like a Gaussian, right? And it's not an isotropic Gaussian, it's an anisotropic Gaussian. The the principal components of that Gaussian, or equivalently the semi-axes of the best fit ellipse to the contours of that Gaussian, are shown with this like oval here, this yellow oval. So it's an anisotropic Gaussian. But as the time step increases, you see the the distribution shift and become more uh, correlated. So what that tells you is that the C component, the observation noise, kind of looks like this Gaussian here, and the variance in the stochastic drive looks like the difference between this Gaussian and this Gaussian. It's that correlated thing giving rise to this diagonal slant right here. So that's the, I don't know, that's the takeaway that you can just eyeball directly from the data in this particular scenario, looking at iron versus MCH. And and what that does is strongly indicate that there is some, maybe I'll skip this slide for now, that there is some, going back here, yeah, that there is evidence for some kind of model of this kind to fit that data, where capital A is a matrix that has off-diagonal components corresponding to this deterministic drive here. And capital B has off-diagonal components that correlate the stochastic drive because this is correlated here. And capital C has some, I don't know, some non-trivial correlation matrix due to this thing here. Okay. And so that's what we can infer for these two variables. And we can go about and do that for every variable pair, and we get these statistically significant correlations. And we can see that they already come in blocks, right? That there's kind of a push-pull relationship between iron and MCH that's we saw before. There's also push-pull relationships. What is going on here? Here, I'm showing so this is an array where every off-diagonal component represents the interaction that you would see in the off-diagonal of A, that the deterministic interaction A, were you to look at a particular variable pair. So let's look at where's iron and MCH somehow. Okay, so here's iron, right? And if you go across iron, you can see MCH right here. So the iron and MCH component is shown here. And what you see is that if you have a lot of MCH, then iron goes down. And conversely, if you have a lot of iron, MCH goes up. Right? So it's this pair right here, this plus minus pair right here, is obtained from that pairwise inference, pairwise fitting. Now, there's one important thing right before I stop. So here are all the statistically significant deterministic interactions. Here are all the statistically significant shared stochastic components, right? Correlated noise. And maybe I can briefly skim over this. These are all not that surprising given the biology of the animals involved, right? All of these components seem to affect each other because they're all related to certain blood-based quantities that you might expect to be related based on biology. And so this was satisfying to the biologists involved because they were able to kind of make sense of a lot of these interactions. And I can talk more about these later. Um, and if you want, we can talk about the age-related interactions. But I just wanted to address one issue, which I think people might be curious about, which is, wait a minute, why are you doing this with pairs, right? Like, you know what I mean? You have this model here. Where'd the model go? You have this model. This model allows for any number of biomarkers. So why don't you throw in all the biomarkers at the same time? Why did you do this pair by pair? And there's a couple of reasons why we did this pair by pair. The first reason is that clinically, you might only have access to a subset of the biomarkers. So if you only have access to two biomarkers, then maybe a pairwise analysis would be more useful to you. But the more important reason why I only looked at pairwise interactions here for this particular time series analysis is that we didn't have an infinite amount of data, right? There was a lot of data, but not an infinite amount of data. And so that meant that the more biomarkers you try to fit at the same time, the more model parameters you have to fit, right? This is just a very basic back of the envelope calculation, right? If you have two biomarkers, A is 2 by 2 and B is 2 by 2. So you're fitting roughly, I don't know, what is that? Eight, not exactly because B is symmetric, but roughly eight things, right? If you have three biomarkers, A is 3 by 3 and B is 3 by 3. Ah, okay. So now you're fitting twice 9. If you have 10 biomarkers, A is 100 by 100 and B is 100 by 100. Ah, so now you're fitting twice 10,000. So the number of parameters in the model scales like the number of biomarkers squared. And so that means that when the number of biomarkers gets large, the number of biomarkers you're trying to fit simultaneously, the power of your data set shrinks. And it shrinks non-linearly, it shrinks superlinearly. And so just as a maybe I don't know, where did it go? What's this thing? Here we go. This is I'm not going to go into the details, but just to you can check this out in the supplementary information of the related paper or whatever. But the basic summary, the TLDR here, is that given the amount of data that we had for the dolphins involved, that was like several thousand measurements and looking at the time distribution, the time increment distribution, and the number of different measurements we had, I tried to do a power analysis, and I concluded that we could pretty reliably back out the OU process for a pair of biomarkers. If that biomarker pair was driven by an OU process, we could back out the coefficient matrices relatively accurately. But if we tried, we didn't have enough data to be able to back out four at a time, certainly. And three was a bit wishy-washy. Three was pushing it, so let's just stick with two. That was essentially the idea. Okay. So anyway, so that was the upshot of this particular relatively short project. And the main takeaway, probably the most important scientific, I don't know, moral from this whole endeavor was this one, which was that clinical biomarkers that clinical blood-based biomarkers that are used time and time again to study humans as well as other mammals are inherently noisy. That if you go to the hospital and you get your blood drawn, and then you go to another hospital on the same day and you get your blood drawn again and have the same test run twice, you're probably going to see some large baseline variation. And that's just one component of the overall variation. The second component coming from an accumulation of unobserved things that might look Brownian if you don't know enough. Okay. So anyway, thanks again. I think that's probably good. Maybe there's some questions.